Welcome to the Mistakes in Living Systems research project. This is a project that comes within a much larger global research program generously funded by the John Templeton Foundation called Agency, Directionality and Function. And that is a global research program bringing together philosophers, biologists and other experts to reimagine, reconceptualize, find new ways of thinking about and testing goal-directedness and related concepts such as agency and function in biology, contemporary biology. The full title of our project is Mistakes in Living Systems, a New Conceptual Framework for the Study of Purpose in Biology. And here we have a picture of a cat and a shrew, not a mouse, by the way, that's important, and also of a blood platelet within the circulatory system. I'll be coming back to those as well. So the team consists of 6 people. There's myself, David Oderberg. I'm the principal investigator or PI for the project, and I'm a professor in the philosophy department and current head of department. We have Professor Jonathan Hill from the School of Psychology and Clinical Language Sciences, also at Reading. The whole team's based at Reading, and Jonathan has a background in medicine and psychiatry with particular interest in mental illness and mental health. Professor Ingo Bojak from the same School of Psychology and Language Sciences at the University of Reading. He is a computational neuroscientist with a background in physics and will be heavily involved in the modelling and experimental work of the project, and as well as all other aspects of it. And the same goes for Professor John Gibbins, who is in the School of Biological Sciences, and he is a world expert on the blood clotting system, which is going to be our target system during the project. And we have 2 postdoctoral research assistants. At the more philosophical end, we have Dr. Christopher Austin. Christopher published a recent book, Essence in the Age of Evolution. And we have François Cinotti, who is more at the experimental modelling end, and he's done modelling work on various kinds of animal behaviour, such as goal tracking behaviour. So our project starts from some fairly intuitive and, I think, plausible assumptions. One is that there are no mistakes in physics. And by that we mean that the objects that physicists study don't make mistakes. Of course, physicists make mistakes. There are mistakes in physics textbooks, and there are mistaken theories in physics, as history shows, etc., etc. But that's not what we mean. We mean there are no mistakes by objects studied by physicists. So no one ever said, or it would be crazy to say that an electron makes a mistake when it goes in this direction rather than that, or that a proton makes a mistake when it's attracted to this particle rather than that. Yeah, the objects of physics just do what they do according to physical law. They can't, an electron cannot disobey the laws of physics. It just does whatever the laws of physics require. But living systems do make mistakes. Biology is special, we think, and different from complementary and supplementary to physics. And by living systems, we mean primarily organisms, but also subsystems like the immune system and also the circulatory system. Parts of organisms as well. But I'll just speak mainly about organisms. Organisms are subject to failure because they're subject to standards of correctness that go beyond the physical. So the standards of correctness for organisms are pretty obvious things like staying alive, first of all, flourishing, being healthy, reproducing, being adapted to their environment. If an organism does something which goes against these kind of standards of correctness, of correct operation or correct function, then things go badly, right? The organism could be unhealthy, sick, starving, sterile, unbalanced in some way, maladapted to its environment, suffering in all sorts of ways, and ultimately can die and will die, as all organisms will. So this kind of generic failure, which you don't get in physics, right? Electrons don't fail, protons don't fail, except relative to some purpose of an experimenter, for example, but they don't fail in themselves, they just do what they do inexorably. But failure of systems in biology is a commonplace and universal, we think. And against that background of general failure, what you might think of as kind of mistake in a broad sense, a mistaken condition for an organism if it's, let's say, starving when it needs food, is a more narrow and philosophically even more interesting conception of mistake, which brings in a notion of agency, that is to say, the kind of failure where an organism gets it wrong. Right, to make a mistake is to get something wrong. So classic example, I add up the restaurant bill incorrectly. Then I've made a mistake, I've got it wrong. If the fish takes the bait, the fish has made a mistake. It's done something wrong. Let's leave aside for the moment whether it knows what it's doing, whether it knows it's made a mistake, whether it could do otherwise, whether the mistake's avoidable in some way. These are all other questions. But the basic kind of action of taking the bait rather than taking what it's supposed to be taking, according to standards of correctness for fish, which is real, edible prey, the fish makes a mistake, and the consequences for the fish of taking the bait are generally pretty bad. Our project is investigating at a very theoretical level and then at a very specialist kind of applied level what it means precisely for an organism or other system to make a mistake. And a mistake in not just the general sense of what we call mere failure, for example, starving to death in the desert, but a mistake in the sense of doing something which departs from correctness, which gets things wrong for that organism in its environment. And think about the kind of language that we have in biology to describe these kinds of mistakes. Being deceived, right? The immune system being deceived by a pathogen that gets around the T cells, for example. Misidentification, organisms misidentify things. Bad timing, doing something too early or too late can have bad consequences for the system. Mislocating something, classic, the dog buries the bone and then doesn't remember where it's put the bone. Quantitative error, right? Error in terms of, organisms doing the wrong amount of something, or getting the wrong amount of food, or the wrong amount of water, or whatever it happens to be. A qualitative error, the wrong kind of thing, like the classic example of the cat and the shrew at the beginning of the slideshow, that picture, the cats don't like shrews, they like mice. Cats jump on mice by instinct, but cats will also generally jump on shrews because shrews look a lot like mice, but they're actually very different to mice, and cats don't like shrews because they give off a bad smell and a bad taste, so the cat will invariably spit the shrew out. The cat makes a mistake, it jumps on the shrew, thinking it's a mouse, makes a mistake, spits the shrew out. That is a mistake if anything else is. If anything is a mistake, that's a mistake. Whether the cat knows what it's doing, why it's doing what it's doing, etc., is a whole other question. And general misregulation of action, not being able to control the way it behaves in some way, is going to be mistaken for an organism. So how might mistake making be modelled? Well, we think it's got to be more than physics and chemistry. Whatever it is that is a way of kind of in an abstract way bringing in mathematics where necessary and other concepts. If you're going to model the making of a mistake, it's got to be teleological in some fundamental sense. It's got to be more than just mere physics and chemistry. There's got to be goal-directedness kind of built into the model, and this is one of the things we're going to be investigating. But our ultimate practical objective is to devise novel, testable hypotheses for working biologists. A model that might be usable by a biologist at the lab bench, or a philosophical framework or conceptual framework that you could use to kind of generate new thinking about biology in a way that latches onto ideas of mistake making that a biologist might not have even thought of before, or might have thought of only in a very implicit way. We want to make that explicit. And in particular, we're going to focus on blood clotting. We have one of the world experts on blood clotting on our team. So we might, this is obviously fairly general at the moment and will become more specific as we go along, but testable hypotheses might revolve around questions such as, can blood platelets misidentify the proteins that activate thrombus formation? So platelets are activated by exposure to collagen, which is given off when there is damage to the skin, for example, and a cut on your skin. Collagen will be released, and the platelets will be exposed to collagen and bind to the collagen, and they aggregate and form a thrombus or a plug to fill up, to block the damage and stop further blood loss. Well, might it be possible for platelets to be deceived? That is, to be fooled into binding or being activated by collagen-like proteins? Can they misidentify what it is that they're supposed to be binding to? And how is the timing of thrombus formation regulated? For example, how do platelets know when to start? How do they know when to stop? How is the sufficiency of thrombus formation determined by the system? So how does the system know, in this case the hemostatic or blood clotting system, know that enough of a clot has been formed to now switch off? Because too much blood clotting is going to lead to ultimately to death. And obviously not enough blood clotting at the right time, in the right place, the right amount, the right quality of clot, not enough of that is also going to lead to death, because the organism can potentially bleed to death. So you don't want too much clotting, you don't want too little clotting. It's got to be just right in the Goldilocks sense. But how is that sufficiency, that adequacy, determined by the system? What kind of things do the parts of the system, such as platelets in particular, do that enable them to regulate clotting? And what kinds of mistake, therefore, can they be subject to? Mistakes of misidentification or some sort of mismeasurement, or wrong decision making, as it were, with decision put in scare quotes, because what does it really mean for a decision to be made by a subsystemic entity like a cell or a platelet? So those are the things we want to look at. And for more, our first article has already been published. That came out in March 2022, and it's called Mistake Making, a Theoretical Framework for Generating Research Questions in Biology with Illustrative Application to Blood Clotting. And that's in the Quarterly Review of Biology on open access, so any of you can download the article, read it at leisure, and let us know what you think, and have a look at the website. And if you are interested in finding out more about the project as it develops, you can sign up for our newsletter via the website and keep up to date with various events that will be happening, and publications and conference presentations and so on. So we hope that you find this project of interest, and look forward to adding more information to the site as the project progresses. Thank you.